This is Dr. Mitch Robinson, and this video is on gluconeogenesis, and it accompanies handout number 10 for cellular molecular medicine. In this video, we'll examine the pathway for glucose synthesis through gluconeogenesis. Gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors. It is one of the two main mechanisms the body uses to keep blood glucose levels from dropping too low. The other means of maintaining blood glucose is through the degradation of glycogen. We need a constant source of glucose in the blood because the brain depends on glucose as a source of fuel. Other tissues use glucose exclusively, and they include red blood cells, kidney medulla, lens, and the cornea of the eye. The daily glucose requirement of the brain in a typical adult is about 120 grams, and that accounts for most of the glucose needed by the whole body during the day. We can use glycogen as a source of glucose, but glucose stored in the body as glycogen is only sufficient to meet the requirement of the body for about 1 day. So, during a longer fast, we need an alternate source, and we make glucose through gluconeogenesis. The figure on page 2 shows how gluconeogenesis fits into the big picture of metabolism. Gluconeogenesis is the reverse pathway of glycolysis. So here 2 molecules of pyruvate are being converted to glucose. And you can see that there are many precursors for glucose synthesis, shown in blue, and they all enter the pathway for glycolysis and can be used as precursors for glucose synthesis. The precursors include amino acids, glycerol, and lactate. Now, gluconeogenesis occurs only in the liver and renal cortex. This pathway involves both cytosolic and mitochondrial enzymes. Although we say that gluconeogenesis is the reverse pathway, some of the enzymes are different. Recall that there are 3 irreversible steps in glycolysis, and these need to be bypassed in gluconeogenesis. These steps are listed on page 3 of your handout. Let's begin with pyruvate. We cannot reverse the pyruvate kinase reaction since it has a very high delta G0. Instead, pyruvate is transported into the mitochondria and it's carboxylated to form the 4-carbon oxaloacetate. Now, this is the pyruvate carboxylase reaction that we've already seen in the TCA cycle. It was the anaplerotic reaction used to make intermediates for the TCA cycle. Oxaloacetate can be converted to phosphoenol pyruvate. But first, we need to take the molecule out of the mitochondria. And as we've seen, oxaloacetate cannot be transported across the intermitochondrial membrane. So, oxaloacetate is converted to malate by malate dehydrogenase, and the malate is transported to the cytosol, where it's converted back to oxaloacetate by a cytosolic form of the malate dehydrogenase enzyme. Now, this transport mechanism has the effect of removing an NADH from the mitochondria and generating an NADH in the cytosol. This is important because we must maintain a balance of oxidized and reduced NAD in the cytosol. So the NADH that's generated by the malate shuttle will be used in the glyceraldehyde 3-phosphate dehydrogenase reaction. Now that we have oxaloacetate in the cytosol, it can be converted to phosphoenol pyruvate. The enzyme is phosphoenol pyruvate carboxykinase, abbreviated PEPCK. This is a decarboxylation reaction, and it requires the hydrolysis of GTP molecule to GDP. So the next irreversible step that must be bypassed is phosphofructokinase. The PFK reaction was driven by ATP hydrolysis, so we can reverse this reaction simply by hydrolyzing the phosphate, and the enzyme is fructose 1,6-bisphosphatase. The final bypass is glucokinase, and again it's bypassed by phosphatase, glucose 6-phosphatase. Glucose 6-phosphatase is found almost exclusively in the liver and kidney. Other tissues cannot convert glucose 6-phosphate to glucose, and so they cannot release glucose into the blood. This enzyme is located in the endoplasmic reticulum of the cell, not the cytosol, and this is thought to be necessary to prevent the glucose 6-phosphate that's formed when glucose comes into the cell from being immediately dephosphorylated. So now we have a mechanism to take two molecules of pyruvate and convert them back to a molecule of glucose. This process of synthesizing glucose is energetically costly. This diagram on page 5 shows that a total of 6 high-energy phosphates are used to generate each glucose molecule. 2 in the pyruvate carboxylase reaction, 2 in the PEPCK reaction, and 2 in the phosphoglycerate kinase reaction. As I mentioned previously, there are 3 sources of carbons for glucose synthesis. Now let's look at those in a little more detail. Lactate is an important source of carbons for glucose synthesis. Muscles produce lactate under anaerobic conditions when they lack enough oxygen to convert glucose to CO2. Lactate is released into the blood, it's returned to the liver, and it can be converted to glucose through gluconeogenesis. The glucose can be resupplied to the muscle tissue for energy production. And this process is known as the Cori cycle. The Cori cycle helps working muscle maintain glucose supply, and it removes the lactate from muscles through the circulation to the liver. Second important source is amino acids. Amino acids are derived from proteins of the diet, and during starvation they may result from the breakdown of body proteins. Many amino acids can be converted to TCA cycle intermediates, and these can be used for the net synthesis of glucose. These are referred to as glucogenic amino acids. All the amino acids except leucine and lysine can be used to generate glucose. Dr. Johnson will go into more detail on the catabolism of amino acids. I'll mention that alanine is a particularly important source of carbons for gluconeogenesis. The alanine cycle is similar to the Cori cycle in that muscle and liver exchange metabolic intermediates. In the alanine cycle, pyruvate is converted to alanine through transamination rather than being converted to lactate. The alanine is transported from muscle to the liver, where it's deaminated to regenerate pyruvate, which is then used to synthesize glucose. Glucose is returned to the muscle for the generation of ATP. This cycle results in the net movement of amino groups from the muscle to the liver, where they can be removed from the body by way of the urea cycle. This is important when muscle proteins are being used as a source of energy. Glycerol is produced during the hydrolysis of triglycerides during fat degradation, and glycerol can be used as a precursor for glucose through gluconeogenesis. Now, the fatty acids produced from triglycerides cannot be used for a net synthesis of glucose. The net product of the oxidation of fatty acids is acetyl-CoA, and our 2 carbon compounds cannot be used to make glucose. Now, there are 2 enzymes that are needed to convert glycerol to an intermediate in glycolysis that can be used for glucose synthesis. Glycerol kinase converts glycerol to glycerol 3-phosphate, and glycerol 3-phosphate dehydrogenase converts glycerol 3-phosphate to dihydroxyacetone phosphate. So DHAP is an intermediate in glycolysis, and it can now be used to make glucose. So remember the 3 possible sources of glucose: lactate, amino acids, all but leucine and lysine, and glycerol. So now let's talk about the regulation of gluconeogenesis. You'll recall that gluconeogenesis occurs predominantly in the liver, and the rates of glycolysis and gluconeogenesis in the liver are controlled mainly in response to the blood glucose levels. Now, we've been talking about the regulation of pathways primarily by molecules inside the cell, and mostly with regard to the energy balance in the cell. But in the liver, gluconeogenesis and glycolysis don't function primarily as energy-producing pathways, but in the storage and release of glucose for use by other tissues. So the regulation is at the level of blood glucose, and the signal is from outside the cell, from hormones in the blood that regulate glucose levels. There are 2 main points of regulation for gluconeogenesis and glycolysis, and that is phosphofructokinase and fructose 1,6-bisphosphatase. When glucose is low in the bloodstream, we want to activate gluconeogenesis, and so we activate fructose 1,6-bisphosphatase. If blood glucose is high, we want to store glucose. Phosphofructokinase is activated. Remember that in liver, glycolysis acts as an anabolic pathway. It's involved in fuel storage. The glucose brought in will be stored as glycogen or converted to acetyl-CoA for fatty acid synthesis. Fructose 2,6-bisphosphate is a major regulator of gluconeogenesis and glycolysis in liver. This molecule is formed from fructose 6-phosphate by phosphorylation at carbon 2 rather than the 1 carbon, as in the glycolysis pathway. Fructose 2,6-bisphosphate is a potent activator of phosphofructokinase, and it's a potent inhibitor of fructose 1,6-bisphosphatase. Increased blood glucose causes an increase in fructose 2,6-bisphosphate. This increases the activity of phosphofructokinase, which activates glycolysis to convert glucose to fatty acids. Now, when glucose is low, fructose 2,6-bisphosphate is low, PFK is inactive, and fructose 1,6-bisphosphatase is active. Now, gluconeogenesis is active and glucose is being synthesized and exported to the blood. So now we know that the level of fructose 2,6-bisphosphate in liver cells controls the rate of gluconeogenesis and glycolysis by regulating the two important enzymes in those pathways. But how is the level of fructose 2,6-bisphosphate controlled? Well, it turns out there's an enzyme known as the bifunctional enzyme that regulates the level of fructose 2,6-bisphosphate. It's called bifunctional because it has two activities. It can both synthesize and break down fructose 2,6-bisphosphate. These reactions are shown on page 8. Now, this enzyme is regulated by phosphorylation, dephosphorylation. If it's dephosphorylated, it has the activity of making fructose 2,6-bisphosphate. If it's phosphorylated, it breaks down fructose 2,6-bisphosphate. These activities, the phosphorylation and dephosphorylation, respond to the hormones glucagon and insulin. Okay, so when the level of glucose in the blood is low, the hormone glucagon signals phosphorylation of the bifunctional protein, and when it's phosphorylated, it breaks down fructose 2,6-bisphosphate. When fructose 2,6-bisphosphate goes down, the level of gluconeogenesis is activated. So low blood glucose results in increased gluconeogenesis. And the opposite occurs with the insulin when blood glucose is high. Insulin rate is increased when blood glucose goes up, and insulin simulates dephosphorylation of the tandem enzyme. When it's dephosphorylated, the enzyme makes fructose 2,6-bisphosphate. This turns on phosphofructokinase and activates glycolysis. So insulin stimulates glycolysis in response to increased blood glucose. Now on page 9 of your handout is a summary of the regulation of glycolysis and gluconeogenesis. And I should point out that there's an additional long-term regulation that we haven't mentioned. After a period of fasting, expression of enzymes needed to synthesize glucose increases. In particular, phosphoenol pyruvate carboxykinase activity is increased by fasting. This appears to have a significant effect on the rate of glucose production. Okay, so that's it for our video on gluconeogenesis.